Hi there, let's pretend we are interested in mycorrhizal fungi in this subtropical forest here. What would we do? Well, first of all, I'm not sure if there actually is that much known about mycorrhizae in this forest here. I haven't found anything, but I haven't looked very hard, so it's possible that I've overlooked something. First step actually would be to do a proper literature search, and then maybe if there's nothing known for this particular island here in Okinawa, maybe you can find information about ecosystems that are very similar and then make some inferences from that about this here. Well, then also, as it comes to mycorrhiza, there's different types, and you can actually find out quite a bit about what mycorrhizal types you are likely to find here about looking at the plant families. So up there you, for example, see some pine trees. They're everywhere here. They're native, evidently, and so pine trees tend to be ectomycorrhizal. So I think that's a pretty sure sign that you're gonna find some ectomycorrhizal fungi here, most likely. Also, some of the trees are, I think, in the beech family, so they also tend to be ectomycorrhizal. But there's also a bunch of other trees here in tropical forests, often are vascular mycorrhizal. And you also find some ferns that tend to be often a vascular mycorrhizal as well. So by knowing something about the plants, you can actually infer quite a lot about what mycorrhizal types you might find here. Also, if there's orchids, which I think there are, I'm not sure about right here, but generally speaking, then you will also have orchid mycorrhiza. Yeah, so this is what you can infer from the plants, but you also can directly look at that. So what we would do is we would just get down and collect some soil. You can take a soil core, for example. So get down here, grab some soil, and then you can pick out the roots basically from that soil core here and take the roots out, stain them following a particular protocol. And you could also just look at the roots unstained and see if you can see signs of ectomycorrhiza, which is typically quite visible because you can see the mantle of the fungus and some of the roots, which I bet you will find here. But if you are looking for a vascular mycorrhiza that is not visible from just looking at the root, you need to clear the root with KOH, and you need to stain it with a stain like trypan blue. You need to make a squash preparation, and then you look at it in the microscope. And then if you see the typical structure like the hyphae, the vesicles, the arbuscules, those are the symbiotic exchange organs and structures that the fungi form inside of the root, then you will know for sure that you have a vascular mycorrhizal fungi here. You can also do that a bit more controlled. You can trace roots to particular species, and so then you get species level data on mycorrhizal associations. Mind you, this is not so easy because roots are typically full of fungi and only some of them are the mycorrhizal fungi. So you need a bit of training and experience in order to recognize them. Well, and what you can finally do is what we do as well. You would take some of that soil, you would extract DNA from that soil, and you would send it to a sequencing facility where you've used the right primers that amplify, for example, all fungi, or specifically just a vascular mycorrhizal fungi. And then you would get a whole another set of data. Namely, you would get basically the phylotypes or species list of a vascular mycorrhizal fungi or other fungi in that particular ecosystem. Yeah, so these are the options. You can also do other things. You can extract hyphae from the soil directly with sort of a filtration method and examine those hyphae after staining in the microscope. But that's much more difficult to learn something about the fungi because the hyphae look pretty much all the same. I mean, there are some distinctive features about the vascular mycorrhizal fungal hyphae. You can differentiate it from others, but that doesn't lead you very far. Yeah, and so currently the best tool we have is high throughput sequencing. You get hundreds of thousands or millions of sequences depending on the method, and then you can basically reconstruct a picture of what the fungal community in this forest would look like. Annoyingly, what doesn't work so well for fungi is metagenomics, which is a technique of choice that people use to learn something about the community composition, but also the function of bacteria and archaea. But that method right now doesn't work very well for fungi. So basically, more or less the best we can do is high throughput sequencing and then learn something about the community composition. Of course, that is not for a casual stroll in the forest. That is actually would be a research project, but to just give you a perspective of what we would do if we were interested in learning more about this place. And with that, see you later. Bye.